In 2010, Sub-Saharan Africa accounted for approximately 68% of the worldwide population living with HIV. The region also accounted for 67% of the world's AIDS-related deaths. This is because of the limited access to reliable diagnostic testing, which then leads to misdiagnosis, inadequate treatment, and increased mortality. In South Africa alone, testing for HIV is available in only 56% of its healthcare facilities. In a region where nearly half the population live on less than $1 a day, the challenge remains to develop a better diagnostic test that is low cost, easy to use, portable, and not dependent on electricity. When a person is infected with a disease or pathogen, the body naturally produces proteins called antibodies to detect the pathogen's toxins, called antigens, so the body can send T cells to fight it off. Thus, infectious diseases can be diagnosed by testing blood for the presence of those antibodies that detect the specific antigen of interest. Our goal is to design a low-cost and accessible diagnostic test based on density. This test, the Mass Density Linked Immunosorbent Assay, or the DeLISA, is not only low-cost, portable, and not dependent on electricity, but it also presents results as a change in levitation height, which is better than a change in color or fluorescence like the standard DeLISA because it promises more objective results. Most importantly, it can detect for the presence of multiple pathogens at once. So not only could we diagnose HIV, but we could also diagnose other infections common in Africa, such as malaria or tuberculosis, at the same time. This assay works by taking small polystyrene beads, adsorbing antigens of the pathogen to its surface, and incubating those beads in the serum being tested. If the serum is positive, specific antibody from the serum will bind to the beads. To detect these binding events, we then bind gold-labeled secondary antibodies to catalyze the deposition of silver from a solution of silver ions. If the person is negative for the disease, the serum does not contain the specific antibodies. Thus, the gold-labeled secondary antibodies do not bind and silver does not deposit. The density of the bead correlates with the amount of silver deposited on the bead due to antibody binding. So we take the beads and insert it in a magnetic levitation device. In this device, the bead's density translates to a change in levitation height. The denser the bead, the further it sinks down in the cuvette. Therefore, the sinking or floating of the beads due to its density tells us if a sample is positive or negative for a disease. With more testing, the DeLISA could bring about great progress in diagnostics and potentially improve the health of many around the world.